Imagine a condition that affects, what, over 12 million people across the globe? It's essentially a mechanical failure inside your heart. The main door, the aortic valve, it just slowly turns to stone.
It calcifies, it narrows, and eventually it just fails. It's a grim reality. We call it aortic stenosis, or AS.
And it is, I think, arguably one of the biggest unsolved puzzles in cardiology.
Oh, it really is.
And the really terrifying part, there is zero preventative medication.
Zero.
No pills, no statins that have been proven to stop it once it gets going. The only option is, well, open heart surgery or a valve replacement.
And that's usually when you're already in dire straits. It's a massive therapeutic gap. We fix it when it breaks, but we don't know how to stop it from breaking in the first place.
But today, we are unpacking a paper that might just change that map entirely. We're doing a comprehensive sentence-by-sentence analysis of a new study in Nature Genetics.
This is Kanasi and colleagues 2025.
Right. And their mission was to see if we can predict this deadly disease by looking at the genetics of perfectly healthy people.
It sounds counterintuitive, doesn't it?
It really does.
Is studying health to understand disease, but that is the core hypothesis here. Can we find the genetic blueprints, the early warning signs, decades before the valve actually fails?
And to do that, they use what I'm calling a technological triad.
I like that.
They combined deep learning AI, genome-wide association studies, or GWS, and then this statistical powerhouse called MTDEC.
It's a massive undertaking. So let's unpack this, starting with the data itself, because they didn't just look at medical records.
No, they looked at images.
Actual images.
They went into the UK Biobank, this huge repository of health data, and they pulled something like 60,000 cardiac MRIs.
Right, from participants who aren't necessarily heart patients. They're just, people in the study.
Which brings us to part one of this whole thing, the healthy valve and this concept of the endophenotype.
Okay, yeah, endophenotype.
That's a $10 word. We need to break that down completely.
Let's do it. So think of a phenotype as the disease itself. Like, yes, you have aortic stenosis.
A binary yes or no.
Exactly. An endophenotype is a measurable quantitative trait that lies on the biological pathway to that disease. So instead of asking, is the valve broken, the researchers asked, how well does the valve function right now?
So they're not looking for a diagnosis, they are measuring performance.
Precisely. And they used a UNet-based deep learning model to do it.
This is the AI.
This is the AI. It performs something called semantic segmentation. It looks at the MRI video of a beating heart, and it automatically outlines the ascending aorta, the main pipe leaving the heart frame by frame.
I love the analogy we were talking about earlier. It's like a computer vision system watching a highway. It doesn't just say there's a traffic jam, that's the disease. It measures the exact speed of every single car to predict that traffic jam hours before it ever happens.
That is a perfect way to visualize it. So the AI extracted 3 specific metrics from these 60,000 healthy people.
Okay, what were they?
First, aortic valve area, or AVA. Basically, how big is the opening?
Measured in centimeters squared.
Correct. Second, peak velocity. How fast is the blood shooting through that opening?
In meters per second.
And third, the mean gradient, which is the pressure difference across the valve.
Right, in millimeters of mercury.
Exactly. And the whole goal was to establish that these numbers, even when they're inside the normal range in healthy people, are heritable, that your genetics play a role in your valve's function long before you ever get sick.
Okay, so they have the AI data, they have the metrics. Now comes part 2, the genetic explosion.
Yes.
They ran a standard GWAS, a genome-wide association study on these metrics. What did they find right out of the gate?
So before they even brought in the heavy statistical artillery, they found 90 genetic loci.
A locus is just a specific location on a chromosome.
Just a spot on the map.
Yeah.
And they found 90 of them associated with these valve metrics.
Were there any familiar faces in that crowd of 90?
Oh, absolutely. They found LPA, which codes for lipoprotein, a very, nasty type of cholesterol carrier.
We've talked about that one before.
Many times. They also found PEL-MD and ALPL, but here's the kicker.
Okay.
Of the 23 genetic locations we already knew were linked to aortic stenosis as a disease, this study found 6 of them just by looking at healthy valve flow.
Wow. That proves the hypothesis then, doesn't it?
It really does.
The same genes that are driving normal variation are the ones that eventually down the line drive the disease. It's a continuum.
It is. But 90 loci wasn't enough for them. They wanted more power. So they used MTG multi-trait analysis of GW.
This is the third part of that technological triad. How does MTAG work?
So imagine you have two maps. One map shows you where all the potholes are on a road. That's the MRI data of valve flow.
Okay.
The other map shows you where cars have actually crashed. That's the medical records of people with a diagnosis of aortic stenosis.
Right.
MTGEC mathematically combines these two data sets. It uses the correlation between the continuous trait, the flow, and the binary trait, the disease, to boost the signal and find more locations.
So they combine the 60,000 MRI scans with a huge meta-analysis. Something like over 1.5 million control patients and 40,000 disease cases.
And the results were, well, you called it an explosion. They were explosive. MTTAG increased the number of identified loci from 90 to 273 overall.
That's repooling.
Basically. And when they filtered for unique distinct signals, they found 166 unique loci across the board. And specifically for aortic stenosis risk, 134 loci.
That is just a massive leap in resolution. Okay, before we get into what these genes are actually doing, let's do a quick recap.
Good idea. There's a lot here.
Okay, first, the problem. Aortic stenosis has high mortality and, crucially, no drug treatment.
Second, the method. They use deep learning on 60,000 MRIs to measure valve flow in healthy people, not just sick ones.
Third, the key insight. They prove that normal valve genetics overlap heavily with disease genetics.
And fourth, the boost. Using MTAG to combine that MRI data with disease records tripled their genetic discovery yield, giving us 166 unique spots on our genetic map to investigate.
And here's a question for you to chew on as we move to the next section. If normal flow predicts disease, could we treat bad flow 20 years before it ever becomes a diagnosis?
That is the billion-dollar question. And to answer it, we have to look at what these genes are actually doing. Which brings us to part 3. The usual suspects.
And I'm going to guess the usual suspects refers to lipids, cholesterol.
You guessed it. One of the strongest findings in this paper was the massive overlap between aortic stenosis and coronary artery disease.
Really?
In fact, 1/3 of the genetic locations they found are shared between the two conditions.
So we're seeing genes like PCSK9, LDLR, LPA, APOE. I mean, these are the heavy hitters of cholesterol metabolism.
They are. But the researchers went a step further. They used a technique called Mendelian randomization, or MIS-CRR.
Nature's randomized clinical trial.
Exactly. Because you're born with your genes, they come before the disease. So if a gene that raises your cholesterol also raises your risk of valve disease, we can be pretty confident the cholesterol is causing the disease, not just correlated with it.
So what did the MR analysis show for lipids?
It provided a really crucial distinction. They found causal evidence that higher levels of APO and LPA, the bad stuff, directly lead to smaller valve areas and higher blood velocity. They cause the damage.
Okay, but what about the so-called good cholesterol, HDL, ApoA?
Nothing. No causal association.
That is a huge clinical pearl. So raising your good cholesterol won't save your valve.
It seems not. You have to lower the bad stuff.
Okay.
It implies that aortic stenosis is, at least in part, a lipid-driven disease. And this suggests that aggressive early lipid-lowering targeting ApoB and LPA might actually prevent the valve from turning to stone in the first place.
Okay, so lipids are a big driver. But this paper found things that surprised me. Let's move to part 4, the surprises. And the first one involves phosphate.
Yes, phosphate metabolism. This was fascinating. They identified a gene called FGF23.
Fibroblast growth factor 23.
That's the one. Now, there's a specific variant here, the C allele at a particular location. If you have this variant, you tend to have higher serum phosphate levels, and you have a higher risk of aortic stenosis.
Okay, so high phosphate equals bad valve. Simple enough.
But here's the nuance, and this is why this paper is so brilliant. They found this link in the disease diagnosis data, but they did not find it in the healthy MRI metrics.
Wait, explain that. The gene predicts the final disease, but it doesn't predict the flow speed in a healthy person.
Exactly. So the interpretation is that phosphate likely drives the progression of the disease, specifically the calcification process, rather than the initiation.
Oh, that makes so much sense. So the lipids might start the fire, but the phosphate throws gasoline on it later to make it calcify.
That is the leading theory. It suggests that once the valve is already a bit damaged, that's when phosphate regulation becomes critical.
Interesting. Okay, another surprise was blood pressure.
Yes, systolic blood pressure. The Mendelian randomization showed a clear causal link. Higher systolic BP leads to higher peak velocity and higher AS risk.
Just mechanics again, pure physical stress.
It is. It's mechanical battering. The valve is slamming shut under higher pressure millions and millions of times a year. That stress damages the tissue.
Which kind of leads us to the third surprise, inflammation.
Right. This validates a long-standing theory that aortic stenosis isn't just passive wear and tear. It's an active inflammatory process. They found genes like IL-6R, the interleukin-6 receptor, and TRF1.
So the valve is inflamed. Is it sensing all this stress?
It is. And this is the fourth and maybe the most sci-fi element here, mechanobiology.
Okay.
They found genes like KLF2 and PKN2.
KLF2 is the shear stress sensor, right? It feels the flow of the blood.
It does, and PKN2 is activated by a mechanoreceptor called piezo-1. So imagine the cells on the surface of your valve. They aren't just passive tiles. They have these little sensors. They can literally feel the blood rushing past.
So if the flow is turbulent, which means high velocity, which we measured with the AI, these sensors get triggered.
That's the idea. Piezo-1 senses the flow, it activates PKN2, and that triggers a whole cascade that can lead to inflammation or calcification. The valve is actively reacting to its environment.
That's incredible. So it's not just a door. It's a smart door that breaks itself if you slam it too hard.
Yeah.
Let's do a quick recap of these mechanisms before we get to the prediction score.
Let's do it. First, lipids, LPA, and ApoB cause the damage. ApoA does not.
Then phosphate. The FGF23 gene is linked to disease progression, the calcification, but maybe not the initial changes in blood flow.
Third, blood pressure. High blood pressure physically batters the valve, causing disease.
And finally, mechanobiology. The valve senses the blood flow using genes like KLF2 and piezo-1, and it actively responds to that stress.
Perfect summary.
Okay, so we have the what and the how. Now for the when. Can we actually predict this in a real patient? Part 5, the polygenic score.
This is where we move from biology to clinical application. The researchers took all this genetic data they got from MTAG and built a polygenic score, or PGS.
And they didn't just test it on the original group, right? They went external for validation.
They did. They validated it in the All of Us research program and the Mass General Brigham Biobank. So real-world populations.
And the numbers, what did they find?
The numbers are striking. Participants who were in the top 5% of genetic risk had a hazard ratio of 3.32.
3.32. So what does that mean in plain English?
It means they are more than 3 times as likely to develop aortic stenosis as the rest of the population.
Wow. And to put that in perspective, that's a stronger predictor than many of our traditional clinical risk factors.
It is. And remember, this is a score you're born with. You could theoretically calculate this at birth.
They also found that the score specific to aortic stenosis worked the best. But even a score based just on the mean gradient, that MRI pressure measurement, was a strong predictor.
Right. It had a hazard ratio of around 2.79. It just reinforces that the genetics of flow are the genetics of disease.
So if I have this high genetic risk, where exactly is the problem happening? Is it my heart muscle?
That's part 6. Tissues and cells. And the answer is no. It is not the cardiomyocytes, the muscle cells.
Not the
muscle. Not the muscle. Using single-cell RNA sequencing, they tracked where these genes are most active. And the guilty parties are the fibroblasts, specifically the valvular interstitial cells and the endothelial cells.
So the structural cells, the scaffolding and the lining of the valve.
Exactly. The genes are highly expressed in arterial tissues, like the aorta and the coronary artery. It confirms this is a disease of the vessel wall and the valve structure, not the heart pump itself.
Okay, before we wrap this up with the implications, we have to be good scientists and talk about the limitations. What should we be cautious about?
Well, the biggest one is population bias. This study was based on the UK Biobank, which is about 95% European ancestry. And genetic risk scores are notoriously difficult to transfer across different populations, so we desperately need validation in more diverse cohorts.
And there was another bias, immortal time bias. What's that?
Right. To get an MRI in the UK Biobank, you had to be healthy enough to survive to the age of enrollment and, walk into the scanner.
So the healthy cohort is inherently healthier than the general population.
It is. And finally, causality.
Right.
Mendelian randomization suggests causality, and it's very strong evidence, but it is not a randomized controlled trial. We haven't proven that lowering phosphate reverses valve disease in a human. We just know the genetics suggest it should.
Got it. Okay, let's do our final recap.
Let's. Prediction. A polygenic score can identify the top 5% of people who are at more than 3 times the risk of developing aortic stenosis.
Target cells. The disease happens in the fibroblasts and endothelial cells, the actual structure of the valve. not the heart muscle.
And limitations. The data is Eurocentric. It really needs broader validation before we can think about rolling it out globally.
So let's synthesize this whole journey. We started with an AI looking at pictures of healthy hearts. From there, we found 166 genetic markers. We figured out that lipids, blood pressure, and phosphate are mechanistically driving the valves to calcify. And from all that, we built a score that can predict it.
It's a real tour de force of modern medicine.
So what? I mean, why should the listener care?
Because it implies aortic stenosis is preventable. For decades, we thought of it as inevitable aging. Oh, grandpa's valve just got stiff. This paper says no. It says it's driven by lipids, it's driven by blood pressure, it's driven by inflammation.
So if we screen someone and find they're in that high genetic risk group, that top 5%, and we also see they have high LPA at age 25.
We might be able to intervene right then. Aggressive lipid lowering, strict blood pressure control. We could potentially prevent the stone valve from ever forming. We could make aortic stenosis a rare disease.
That is an incredibly hopeful future. But it does leave me with a heavy question for you and for our listener. Knowing that these normal variations in your biology can predict a deadly disease decades down the road. Would you want to know? Would you want a polygenic risk score for your heart valve printed on your chart at age 20? Or is knowing your fate just a burden?
It's the classic dilemma of genomic medicine, isn't it? Knowledge is power, but only if you can actually change the outcome. I'd like to believe that with this research, we finally have the tools to start changing the outcome.
A fascinating thought to end on. Remember, this was a deep dive into the source material and definitely not medical advice. Thanks for listening.
Stay curious.